Hello and welcome to this video tutorial. My name is Dr.Anjusha Mune, Assistant Professor, Sage University, Bhopal, Sage School of Sciences. And the topic of this video tutorial is nucleotide composition calculation using MEGA. Nucleotide is the building block of nucleic acid. Nucleic acids are naturally occurring chemical compounds that serve as the primary information carrying molecule in the cell. They play essential and important role in directing protein synthesis. There are two main classes of nucleic acid, that are deoxyribonucleic acid DNA and ribonucleic acid RNA. DNA and RNA are polymers that are made up of long chains of nucleotide. Nucleotides consist of sugar molecule, either ribose in RNA and deoxyribose in DNA, that is attached to phosphate group and nitrogen containing base. This base have adenine, cytosine, guanine and thymine in DNA, and in RNA the base uracil takes the place of thymine. In order to understand the genome in a better way, it is important to understand the frequency of occurrence of each nucleotide in a genome. The composition analysis, we calculate the overall frequency of occurrence of nucleotide adenine, cytosine, uracil and guanine, and the frequency of each nucleotide at first, second and third site of the codon will also be calculated. And to perform this experiment, we will use an online available tool, freely available tool, that is MEGA. The downloading and installation of MEGA software we have discussed in our previous video. So after the downloading of the software, what you will do, open the software and you will see a terminal like this. In this terminal, on the top panel, the third option you can see it is written as model. When you click on that option, a drop-down menu will open. There, select complete nucleotide composition, compute nucleotide composition option. When you click on that option, it will take you to another page from where you can select the file for which you want to perform nucleotide composition analysis. The file can be a text file or sequences in FASTA format or a multiple sequence alignment file, or you can also calculate nucleotide composition directly for a sequence by retrieving the sequence from a database. The software is connected to database. You can directly import the sequence from database also. So it depends upon the type of data you are using to calculate nucleotide composition. When you upload the file, it will provide you option protein coding nucleotide sequence data. You click on the yes button. After that, it will ask you about the type of output you want from the analysis. There are various output that MEGA provide. So when you click